Okay, so we are finishing up chapter 27 on prokaryotes. Okay, and we've already talked about their structure, their diverse metabolism, their mode of reproduction, which is binary fission, where they're found, which is everywhere, okay, on land, in water, all different environments. Now, and we now need to talk about how they get genetic variation. So if we go back, and we said that because they grow so fast, you can get evolution in real time. Now, in the chapters on evolution, what we talked about was that in a population, you have to have genetic variation, right? You have individuals with varying traits, okay? And how we covered that when we talked about microevolution was we talked about changes in the allele frequencies, okay? Now, in eukaryotes, like humans, we have 2 copies of each gene, okay? So we have 2 alleles for each trait, or each gene. In prokaryotes are haploid. They only have 1 copy of each gene, okay? So we don't use the word alleles quite the exact same way in prokaryotes. Now, prokaryotes do not go through anything like meiosis, okay? They go through binary fission, which is reminiscent of mitosis, but they do not go through meiosis because they are haploid. There's only 1 copy of each gene. There's only 1 chromosome. So you can't have chromosome pairing. You have no such thing as homologous chromosomes. You can't have chromosome pairing. You can't have homologous recombination. This was in Bio 1. We talked about crossover and swapping of genes between homologous chromosomes. Okay, that's called crossover. We also call that homologous recombination. Recombination means recombining or swapping between homologs. So in eukaryotes that reproduce sexually, okay, and we talked about sexual reproduction, right? That requires mating between 2 organisms of different genetics, okay? When we always talked about a sperm fertilizing an egg. In prokaryotes, there is no mating, okay? It is 1 cell giving rise to copies of itself, right? So it's just 1 cell giving rise to 2, 2 to 4, but there is absolutely no mating, no exchange of, no swapping of genetic material. Okay. So, but genetic variation does occur in prokaryotes. Okay. Any living organism evolves. Okay. Genetic changes will occur. Okay. They just occur by very different mechanisms in prokaryotes. In eukaryotes, genetic variation recombination in organisms that mate occurs by meiosis. In prokaryotes, okay, which do no mating, all their binary fission is an asexual reproduction method. Asexual meaning not sexual, no mating, okay? In prokaryotes, where there is no mating, it is completely asexual. The way of getting genetic variation is called horizontal gene transfer, okay? And horizontal gene transfer is sharing or transfer of DNA between unrelated prokaryotes. And I put bacteria up there because I tend to be a little bit bacteria-centric, okay? And I know a lot more about bacteria than archaea, okay? And I know these processes happen in bacteria. Probably in archaea as well. I'm just not as much of an expert on archaea. So the main way bacteria get genetic change is by horizontal gene transfer. And Genetic change is genetic variation and genetic recombination. Another way that bacteria get genetic change is by mutations. Okay? Mutations that occur when DNA polymerase makes mistakes during replication. Okay. I'm going to go over 3 types of horizontal gene transfer. One is called transformation. Okay? And transformation is when bacteria uptake DNA from the environment. So let's say in soil, some bacteria die. When they die, they lyse and all their contents spill out, including their DNA. And then it gets chopped up into pieces. Another bacterium can take some of those DNA pieces up and incorporate them into their chromosomes. And then they've picked up genes and they're genetically distinct. Okay? They've gotten genetic variation by picking up DNA from the environment. That is called transformation. Conjugation is transfer of DNA between 2 bacteria. Okay, so it's direct transfer between 2 bacteria. And if you think conjugation, think conjugal visits. You have the prison conjugal visits, right? Like boom, boom. Conjugation is often called bacterial sex. Okay, and what happens in conjugation is 2 bacteria meet up, and their cell membranes and cell walls fuse, and their cytoplasms are put in contact with each other, and genetic material can transfer from one to the other. Okay. In transformation, you only need 1 live bacteria and DNA in the environment. In conjugation, you need 2. The third is transduction. Okay. And transduction is transfer of DNA from something called a bacteriophage. Okay. And bacteriophage are bacterial viruses. Okay. So there are viruses that are specific to bacteria, and they're called bacteriophage. Viruses specific to animals are called animal viruses. Okay. So for example, SARS-CoV-2 is an animal virus. Okay. Now, bacteriophage, when they infect a bacterial cell, they can transfer DNA. So transduction is transfer of DNA from a bacteriophage to the bacteria. I'm going to show you some pictures to show you these, and these are modes of recombination, okay? Not reproduction, recombination, increasing genetic diversity. Okay. So here is showing transformation, okay? You have a cell, it dies, and when it dies, and just take my word for it, the chromosome gets chopped up into pieces, then it gets spilled out in the environment. Not all bacteria are naturally transformable, but some are, and they have the ability to bind to the DNA in the environment, bring it into the cell, this is a live cell, and then integrate or recombine, insert basically, this piece of DNA into the chromosome. Okay. Notice it's called homologous recombination. And then now the chromosome of this bacterium is changed. It's got additional genetic material that codes for additional traits. Okay. All right, conjugation. Okay, there's some terms here you don't need to know. Okay, like all this F plus, F minus. Don't really worry about that too much. Well, I'll go through it because it'll make it easier. All right, so here's one bacterium. Here's another bacterium, and they make a structure called a sex pilus, okay, which is a appendage, right? A protuberance that's made out of protein, okay? And when one of them will extend its pilus, here they're showing this pilus extend, the pilus will attach to the other bacterium and then retract and bring it closer. So if you extend your arm is your pilus, your body is your bacteria, you extend it out, you grab another person, let's say, and then you go, hey baby, come on over here. You bring them closer, right? But you're using the pilus to do that. Then they're brought closer, and the 2 cells fuse together by what's called either conjugation or a mating bridge. Okay, so here's two bacteria. One contains something called an F plasmid. Okay, so here's a chromosome. So some bacteria have additional DNA that exists on a circle, and that's called a plasmid. Okay, it's just an extra place for genes to be held. Now, here's that pilus. It'll attach to, so this one's called a donor cell. This one's the recipient. The one with the pilus that attaches is donor. The one that gets caught is the recipient. Pull it closer, then you're seeing the cell wall and cell membrane will fuse, and you'll have this bridge. And then this plasmid can get copied, and as it gets copied, it rolls into the recipient cell. Oops. So that means this DNA from the donor cell got transferred into the recipient. So the genetics of this recipient cell got changed. And all the F thing means this plasmid is named an F plasmid. If a cell has it, it's called F plus genetics. If it doesn't, it's called F minus genetics. So as a result of conjugation, this F minus cell gets converted to an F plus cell, okay, because it's acquired additional DNA by conjugation. Transduction. Okay. Transduction. So this is a phage. Okay, it looks like this funky little alien creature. I kind of think they're really pretty cute. It's pretty much all protein and then nucleic acid. Okay, it's a phage's bacterial virus. It's gonna stick on a bacteria. It'll insert its genetic material. Okay. Now there's something that happens when the phage does it. It causes the host bacterium to make copies of the phage genome as well as in addition to the chromosome. And somewhere along the way it'll cause the host DNA to kind of get chopped up a little bit. And then they'll get all repackaged to make more phage particles. So with all viruses, they get into your cell, well not all, but many, they get into a cell, they copy, and then often they have to get out so they can, once they've used all the resources of one host, they can then go use the resources of another host. Okay, so they've got to get out. Okay, so with SARS-CoV, the virus has got to get out so it can infect other people, and how it gets out is through our nasal secretions and our breathing, our respiratory, basically all our nasal and mouth respiratory secretions. Okay, so now these phage get out, but some of the phage have picked up some of the host bacteria along with its own genes. Some host bacteria genes along with its own genes. Now when it gets out, it infects another bacterium. Now this bacterium has a yellow chromosome. When it injects, it may inject some of the blue DNA from the previous bacterium. So it'll pick up DNA from one bacterium, then when it infects a new one, it'll pass it along. Okay, this does not happen in humans, by the way. This is not animal viruses. This is with bacterial viruses, so don't worry about that with humans. It'll inject it, and then that DNA will get incorporated, right? And then you've changed its genetics, okay, using phage. Okay. And that's pretty much that on horizontal gene transfer, which is the main mode of recombination and genetic variation. Okay. I'm going to bring up one other benefit of bacteria. Now, some of you may not be familiar with it, and if you're not, no worry. Okay, there's some relatively new, it is new, gene technology called CRISPR. Okay, some of you might be familiar with it. And CRISPR gene technology allows you to specifically alter a specific gene. Okay, so CRISPR has been used or tested, not widely, very limited, for sickle cell anemia to replace the mutated sickle cell gene with the wild type. Okay, so it's very specific technology, but that technology, and I don't have time to go over it in this class, that technology derived from what bacteria can do. So bacteria basically use this CRISPR mechanism, okay, basically as a means of protecting itself from foreign DNA that enters. Okay. And scientists have been able to figure out how to use this bacterial mechanism, this CRISPR bacterial mechanism, and then apply it to eukaryotic cells. So for CRISPR technology, we think bacterial mechanisms. Now, what most of you are somewhat familiar with bacteria is more the diseases they cause than the benefits. Of course, I focus on the benefits because, well, I really like bacteria, okay? And I really like that they're beneficial. But here's a chart in case you're curious, okay, of various bacteria and the diseases they cause. Okay? Now, obviously, I'm not going over all of this, okay? But you guys have probably heard of the disease anthrax. That's caused by a bacterium, Bacillus anthracis. You may have heard of the disease cholera, and anthrax is a respiratory disease. Okay, cholera is a gastrointestinal disease that has you basically like really, really watery diarrhea. Okay, that often comes about after natural disasters where sanitation infrastructure has gone haywire. Okay, so major killer in times of crowding and poor sanitation, right? Vibrio cholera is a bacteria that causes that. Okay. Cavities are caused by a bacteria, certain bacteria in the mouth called Streptococcus. So specifically Streptococcus mutans. So when you're brushing your teeth, you're removing that species. Let's see. Gonorrhea, sexually transmitted disease, is caused by the bacterium Neisseria gonorrhea. Neisseria meningitidis causes bacterial meningitis, which you might be familiar with. Let's see, you might have heard of Lyme disease. That's caused by Borrelia burgdorferi. You may have heard of the plague, the bubonic plague. That's Yersinia pestis. Okay, you've heard of TB, tuberculosis, caused by Mycobacterium tuberculosis. There's a variety of different bacteria that can cause pneumonia. Okay, so these are just some of them. There are actually more. No. Yeah, these are the main ones. Okay. If you have any questions on prokaryotes, feel free to ask them. Hopefully this was all understandable. And hopefully I've covered everything that's on your study guides. If I missed something, just text me. You can text, email, call, whatever. Just ask the questions, okay?